Our next speaker will be introduced by Michelle Worth.
Sisu Carter is a professor of psychiatry and the co-director of the Brain Body Center at the University of Illinois at Chicago. And previously, she has served as Distinguished University Professor in Biology at the University of Maryland and Professor of Psychology, Ecology, Ethology, and Evolution at University of Illinois Urbana-Champaign. Dr. Carter studies the neuroendocrine basis for pair bond formation, having established the prairie vole as an excellent model for pair bonding. And her research includes effects of the hormones oxytocin and vasopressin on development, a topic that has implications for human health and well-being, including developmental disorders like autism. Furthermore, Dr. Carter conducts research in both voles and humans, thus extending and expanding the findings in voles to human relationships, whereas the vole model allows manipulation and fine-grained analysis of brain mechanisms. So from this powerful synthesis, Dr. Carter has contributed greatly to our understanding of the brain and hormonal mechanisms of social bonding. So there's more to say about all her papers and awards and a number of articles and chapters that she's co-edited. And I've heard her speak before at Michigan. I'm very excited to hear her again. So here is Dr. Carter.
Thank you. Thanks, Michelle. Thank you, and thanks to everyone who's made this very interesting conference happen. Okay. The previous speaker was my husband of 40 years. The fussy baby was one that I nursed for 2 years. You don't get any medals for that, especially when your mother's saying, "Are you still nursing that baby?" But anyway, never had a drop of formula, so whatever causes it, we can't blame on something simple. I'd like to talk, though, today about a hormone that I'm very interested in and sort of the biological basis for how this chemical might have come to affect behavior. And I'm going to argue, and I don't think this audience will need much persuasion, that the evolutionary prototype for social behavior and for social support and social bonds is really the parent-child interaction. And even in a generally asocial species like the orangutan, there are high levels of social behavior with their infants, with their offspring. So at the center of this process is this very interesting chemical that's been implicated in both male and female parental behavior, and it's called oxytocin. So why is it so important? And I'll move backward and say that most of what we know about biology has come from an analysis of individuals. This is also true of psychology, a little bit less so. But the biological fact is that the living organisms, even as far down as bacteria, can do better when they're with others, even asexual organisms. So survival, and especially mammals, vertebrates, reproduction depends on more than one. And in the same process, the mammalian nervous system has developed a kind of dependence on each other, on others, for physiological and behavioral homeostasis. So Myron Hofer called these hidden regulators. We know more about them by what happens when they're not there. When we remove the other, we see an increase in drug abuse, mental dysfunction, substitutions. Oxytocin, I think, is at the heart of how all this is put together. It's a very simple molecule. It only has 9 amino acids, a ring and a tail. And its history sort of got it off the mainstream because it was viewed as a female reproductive hormone. And that really was bad news for this chemical because people thought, well, it's only involved in females. And up until the late 80s, you can find books saying that there's no known role in males. This is not true. We now know that oxytocin is central to understanding the biology of social behavior, social support, sexual behavior, all kinds of processes. And this very nice slide from a paper by Zoe Donaldson and Larry Young shows sort of the evolutionary process here. So we have a primate, mammals with vasopressin and oxytocin, other vertebrates having similar molecules all the way down to invertebrates. So this is not a new compound. It's nothing particularly new, but in mammals, it has a companion, vasopressin, that's very, very similar. You'll see here in blue that there are only 2 amino acids different. And I'll argue this morning that that interaction between oxytocin and vasopressin is quite important to how these chemicals work. And I want to show you one slide that Hert de Vries from the University of Massachusetts has loaned me, which shows you something quite interesting about vasopressin. This yellow is vasopressin staining. These are male rats, these are female, and most species are similar. This staining is up in an area called the lateral septum, which gets its projections from the medial amygdala and beta nucleus of the stria terminalis. 2 brain areas that you all know about because they're part of sort of our vigilance and defensive system. And I'll argue again this morning that mainly the differences between oxytocin and vasopressin, a lot of them are based on the fact that vasopressin has a rather different role in helping protect us from danger and different kinds of environmental processes. Now, my interest in oxytocin started when I was nursing this first baby, 1980. And I began to realize that strange things were happening, things I couldn't find in textbooks to explain to me what was the nature of the process that I was experiencing. So this is research. And what I discovered, and lots of people now accept, is that lactation was a kind of biological basis for social bonding, offered this basis. It was first studied by Barry Cavern in Cambridge, showing that lactation was necessary, not lactation, but the hormones of lactation were necessary for social bonding, and that at the core of that was oxytocin. Now, we're involved in a big experiment, and many of you are very sensitive to that, and that is that historically breastfeeding was the only viable method for nourishing newborn humans. Then the formula came along, and the mother's brain is really changed by not lactating. And I want to briefly tell you that this is the wet nurse, sort of a classic picture of an upper-class lady who did not nurse her babies. But now, especially since World War II, we have a huge sort of change in the rearing of children, and women begin to make decisions about whether or not they'll provide milk through lactation for their infant or not. But most of them don't realize they're making a decision about how their brain functions, and their whole endocrine system is changed by that decision. Of course, the biological prototype, as I'm saying, is really, for mammalian sociology, is really the mother-infant interaction, which we can see very easily. Parenting is not limited to mothers, and it's not limited to biological offspring, but not everyone is prepared to be a parent. And we may need help. We're not just really not all there. So we have probably been given some extremely powerful insurance factors to try to encourage us to show the correct behaviors. And what oxytocin and lactation are somehow doing together is allowing the new mother to manage stress more effectively. I would argue again that it's really a buffer between the extreme hormonal events of pregnancy and the postpartum period. And that, of course, it's normal to lactate. Women who historically who didn't lactate were usually not doing so because their baby didn't survive. So it's a very abrupt physiological change to go from pregnancy to non-pregnancy without lactation. And lactation then provides part of this buffer which allows us to deal better with even physical stressors, but certainly psychosocial stressors. Now, since 2005 mainly, people have begun to study oxytocin using the intranasal oxytocin form that was really originally sort of designed as a lactational aid. And these are just some examples. Intranasal oxytocin improved the ability of male humans to read the emotions of others from pictures of their eyes, and also increased something that a group of neuroeconomists, led really by Marcus Heinrichs at the University of Zurich, showed that even in computer games, a little extra oxytocin allowed people to be more trusting, as measured by how much money they would give away. Endogenous oxytocin, which is made primarily in the brain and released into the blood supply, as we said, can of course facilitate birth. It also allows milk ejection, but it's also released out into the brain, and it's this that we're interested in today mainly. But I want to make one more kind of point here, something that's coming out of the literature and is very exciting, I think, and that is that under normal conditions, the body also uses oxytocin as an anti-inflammatory and an antioxidant. And animal studies show that you can heal burns more quickly. Deliberate damage to the heart is healed by extra oxytocin. Plays a role in breast cancer, a little more complicated than simply making it better. But one of the most interesting findings is osteoporosis, which is going to kill us all. If something else doesn't get you, your bones will go and you won't be able to be upright on the planet. So osteoporosis and bone reformation and sort of keeping your bones healthy is facilitated by oxytocin. Intestinal bowel diseases, there's a role for oxytocin. And stroke, and of course some of the things that people have been more interested in are disorders like autism, schizophrenia, possibly anxiety and depression. I did a study in collaboration with Janice Kekol-Glaser at Ohio State and Jean-Philippe Goyen. Sorry, this is very sensitive. And what we found was that individuals with endogenously high oxytocin were able to heal a wound, a deliberate wound, much more quickly, statistically more quickly, I shouldn't say much. There's also an interesting relationship. This is the work of Leah Rubin at the University of Illinois, one of my mentees. And what Leah found was that there was people with high levels of oxytocin had lower levels of what are called PAN scores, which are symptoms of schizophrenia. So all of the people shown here have been diagnosed as schizophrenic. The ones with the lowest levels of oxytocin tended to have higher symptoms, and this has been repeated. And we've also seen that in schizophrenia, very severe form with polydipsia, the oxytocin levels on average are lower. They're not lower in normal water balance schizophrenics, but people who have a water balance impairment have particularly low levels, in this study at least, of oxytocin. This was Maury Goldman, another collaborator of mine from Chicago. So, what is it? How does this stuff work? How can such a small molecule be so important? And first, where does it come from? I've already told you it's found in the brain. These dark spots are from a vole brain, stained for oxytocin. It comes out of a sort of heart-shaped structure known as the paraventricular nucleus, and also another related area called the supraoptic. The paraventricular nucleus is really at the heart of the stress response axis. It sits right in the middle of the hypothalamus. It has processes going and coming from everywhere, including the periaqueductal gray, very important brain area, but just about everything you can think of. And it's also, what's odd about oxytocin, it's not really a neurotransmitter. It's a neuromodulator, and it flows through the brain. It moves through the brain in kind of what's believed to be volume transmission, meaning a path of least resistance. But what is interesting, the classic model of the HPA axis, which almost never includes, even today, oxytocin in it, is the brain area, the hypothalamic area, including the paraventricular nucleus, contains vasopressin and CRF, both of which are usually thought of as stress hormones. So under a stressful experience, it goes up, ACTH goes up, CORT goes up. You all know this story. But in reality, there's another player, oxytocin, in those same areas, sometimes in the same cells, at least in the same cells as CRH. And in a context of safety, social support, a good time, during good times, we would probably expect the emergence, positive social behaviors, growth, healing, restoration. But interestingly, and somewhat to my surprise, we also see oxytocin released in a context of fear or threat. And acute stressors are probably one of the most powerful ways to release oxytocin. Oxytocin in animals, it facilitates approach, a sense of safety probably. In high concentrations, vasopressin has rather opposite effects, causing avoidance or withdrawal and a sense of, we believe, threat or fear, at least in humans. And when given as an exogenous treatment, the effects of oxytocin and vasopressin tend to be different in males and females. And this is something that so far hasn't really been covered very well here, but I think it's quite important to what we're trying to understand. So we have areas that you've already heard about, periaqueductal gray, brainstem areas that regulate the autonomic nervous system, and we also have within this system the amygdala, with a particularly interesting area called the central nucleus, where oxytocin and vasopressin play out a little dance with each other, in which one seems to facilitate approach, the other avoidance. Now, this is the simplified version of this story, and as said, really, the whole story is much more complex, and these arrows should be going both ways, from the brainstem up and down. So, what's oxytocin? Okay, is it simply a metaphor, a physiological metaphor for safety? Perhaps the tip of a physiological iceberg of interactive systems throughout the whole body, and one which is really only just in the last decade being recognized. Now, my own work has focused on something called the prairie vole, Microtosocrogaster, very small mouse-like rodent, and a lot of people have helped with the work that I'll talk about. I want to particularly acknowledge Karen Bales and Dr. Porges, because I'll talk about a little bit of their data today, and Angela Grippo. And what we have found in prairie voles is that they do form lifelong social bonds. These social bonds are not identical to sexual bonds. In fact, prairie voles and most monogamous species, in most, sexual exclusivity is not the trait that seems to define the animals. It's who they live with. And this, of course, is true of humans, too. It's very important who's with you, perhaps more important than who provided the genetic material for your offspring. Prairie voles exhibit these bonds. We can see also biparental care and young animals taking care of babies. This animal's very carefully moving a baby around. We studied them originally in Nature. Lowell Getz was my collaborator who did the field work and brought them into the laboratory and found right off the bat what I think really got me back to oxytocin. In addition to my children, the prairie vole brought me back because the animals that were mating formed pair bonds more quickly. Now, living together is adequate. They can show social preferences without mating, but mating, which we already knew back in the 1990s, released oxytocin, seemed to be a particularly powerful stimulus for pair bonding between both males and females. It played a role, we later found, and many people, including Alison Fleming, who's here, and many people have studied maternal behavior, but there's also a growing literature on male parental care. The prairie vole really does have high levels of oxytocin, extremely high, and it has an autonomic nervous system that's rather human-like, with high levels of vagal tone. And possibly it's this ability to show parasympathetic activity and to have this influence the heart in a strong way that helps to explain why prairie voles are able to form these social bonds. Oxytocin, then, we know, is released under certain conditions that involve positive social behaviors, but it can also be released when animals are interacting aggressively, and I would suggest perhaps serving as a component of the coping and buffering mechanism. So here are just some data from Angela Grippo's paper, one of the ones that Angela did with us. She was a postdoc that worked with both me and Steve, and what she did was put males and female prairie voles. Some of the animals were isolated, and that's important here. Some were living in pairs, and they were both exposed to a 5-minute intruder, and what happened was that the animals living alone released extra oxytocin, again, possibly taking care of their needs. So this may be a compensation, possibly in allowing these animals to deal with the stressor, at least emotionally. Angela also put we put monitors into the voles and studied heart rate And what you can see is that when animals are isolated over a 4-week period of time, heart rate goes up. If they're living in pairs, it didn't do that. And if they were isolated and given in the last 2 weeks extra oxytocin, they did not show the autonomic effects of isolation. They also didn't show a whole battery of other tests were blocked. Now, we don't totally understand all the mechanisms of this, but I think it's quite important that it at least documents how oxytocin, the power of oxytocin. I was asked to talk about early experiences, and I love talking about this. But this came again from my personal experience and my concern over the effects of early handling and early treatment of babies, and trying to, because I'm a biologist by background, and to me, anything that's natural has to be better than something that somebody made up in 1942 who had never had a baby and wrote as a lot of our traditions come out of things that really weren't based on natural situations. So I began to sort of try to understand this using the prairie vole model and looking at some of the traits. And I won't have time today to show you all the data, and there is no such thing as normal. I don't know, a typical, average, there's everything and every one of us is an adapted organism. But under certain kinds of conditions, the basic model here is that normal amounts of external stimulation act on the parent, possibly to stimulate the offspring in a particular way, which may reduce anxiety and at the same time allow that animal, when it gets older, to be more parental and more social. But this system is not always perfect, and we have to adapt. And one of the mechanisms for adaptation, interestingly enough, what our data are showing is that actually you get more oxytocin, especially in females, under conditions that require adaptation, when there's adversity, and even when you excessively manipulate the parents. And this, in the male, this sort of extra handling seems to actually make the male more anxious, reduces the alloparenting that animal shows in later life, possibly because the male is coping in a different way. He's putting out extra vasopressin. This vasopressin is part of an active coping and vigilant strategy. So, of course, we're involved in this huge human experiment, the largest one in history. There are 2 parts to it. One is that we're allowing the birth process to become a biochemical event with a series of hormones, one of which happens to be oxytocin. And this is, if anything, if I'm going to do anything the rest of my career, my husband tells me I'm a little overly passionate about this, I would like to see us work on a better birth process. That doesn't mean we throw out all the Pitocin, but it would be, it seems almost about time, doesn't it? And I know I'm preaching to the choir here, so we have to get out of this room and on to the bigger world. But we just know so little about the developmental effects of oxytocin that to be using it in such a way, in fact, now it's almost 100% of women giving birth are either given oxytocin to induce, to augment, or if she doesn't get it then, if she misses that opportunity, they'll give it postpartum to reduce hemorrhage. So we're involved in this amazing experiment. In some parts of the world, we're also blocking oxytocin. That's even more scary. And there's been nothing done in any animal model to speak of on the effects of blocking oxytocin to prevent premature labor. Prematurity is a huge medical problem. It's, some people think, the largest medical problem on the planet. And trying to keep women from from delivering prematurely is important. But whether blocking oxytocin is a good idea, I think is really open to question. Also, human milk, as you may know, contains oxytocin. Breastfed babies receive different amounts of handling and attention, so we're just involved in it. And that's almost certainly an oxytocin manipulation. So we're involved in a huge experiment. Nobody signs a consent sheet. No one realizes they're being part of this experiment. About 10 years ago, I began to do studies just giving the oxytocin to the baby. It's kind of unethical to do to animals what we do to humans, so I wouldn't think of that, but believe me. But we could give a single injection of oxytocin to the baby on the first day of life, and we looked at pair bonding and alloparenting later. And I won't go through all this in detail except to show you that the males were particularly affected and particularly impaired by blocking oxytocin on the first day of life. We gave extra oxytocin to some of the animals, and that had also very interesting effects. Let me go through the OTA, the blocking first, momentarily here. If we blocked them, we interfered with the production of vasopressin, not oxytocin, which actually was a little higher, but vasopressin. And anything that interferes with vasopressin has effects for behavior. And we also found the oxytocin receptors were changed. Animals that had either the blocking of oxytocin or extra oxytocin showed effects. And I don't want to go into the details, except I'm going to show you just the outcome of this, which is that changes in the V1A receptor, very important behaviorally and also for blood pressure, that receptor was actually upregulated with a little extra oxytocin, not with the oxytocin antagonist. And especially females actually had less vasopressin receptor binding if they were given extra oxytocin. This was work that Karen Bales and I did years ago. Now, the negative effects of early oxytocin could be reversed of oxytocin antagonist, which is here. So we have very low levels of care of young in animals given extra oxytocin antagonist on the first day of life, one injection. 8 days later, we gave them some extra oxytocin, and in fact, they were restored to normal. So I think this documents a role for oxytocin, but doesn't really go through the mechanisms, which I believe are more vasopressin dependent. In conclusion, oxytocin, which I haven't shown you all the data, programs the nervous system. It causes changes that are sexually dimorphic and also dose dependent. Extra oxytocin, actually very low levels, facilitated male behavior. High doses had all kinds of effects. In females, I'll show you the effects of very high levels. Whoops, I'm going to skip a couple slides because I think our clock is running out. Let me go back because you guys are going to like this. Okay, we started out with the peptide studies. And over time realized that maybe these effects, every little thing we were doing mattered. So Bales and I began to do these studies in which we gave either one handling experience on the first day of life or none, left the animals alone. Well, the animals left alone, which at first we thought were going to be unstressed, were in fact not typical at all. That won't surprise you. And they're, for example, showing alloparenting went down, the behavior went down. And they failed to form a pair bond later, which really was surprising to us because it's very difficult to mess up pair bonding in the prairie vole. But the unhandled animals had atypical behaviors. They did not show the pair bond that's normal of this species after a period of time with another animal. They were sort of random in their choice of partners. We began to study this in a variety of ways, and what we found was that, in fact, not handling them, quite opposite to what we expected, actually increased the amount of oxytocin, number of oxytocin receptors in the nucleus accumbens, which is an area that you'll probably hear about in the next talk from Allison. And in both sexes, there was an increase in oxytocin receptor in the bed nucleus. This increase in oxytocin receptor I'm going to go on to the next. I'm going to skip this piece because I want to tell you something. First, I've got these out of order. Last night I started taking out slides. Bad mistake. And so I put some of them back, and the order doesn't make too much sense anymore. But what I want to say first is that, then I'll have to backtrack just for a minute, is the extra oxytocin given on the first day of life had effects that we at first were surprised by. It actually increased the facilitated pair bonding, but when the dose was higher, it blocked pair bonding. In fact, they went to the strangers. This is a little bit frightening, I think, because we don't know what's the dose. When we're giving it to a mother, we don't know if any of it's getting to the baby, but there's a fair chance it is. It could be passing through the placental barrier. And we don't know what any of that is doing to the receptor because we haven't begun to do these experiments yet. But in the unhandled animals who have atypical social behavior, we have rather high levels of oxytocin receptor. I'm sorry, this is wrong. And we have a change in methylation. Now, everybody has to do epigenetics, and this was done by a wonderful collaborator, Jessica Conley at the University of Virginia, and a medical student working with her, Jim Cronk. And what you're seeing here are DNA methylation profiling of the oxytocin receptor in the nucleus accumbens on a site that promotes the expression of the oxytocin receptor. And what we found, again, completely consistent with the autoradiography and completely opposite to what I would have predicted, is that the animals that did not get handled had less methylation. Less methylation should these result in more oxytocin receptor, and that's what we found. And it was there are 12 sites. This is called a CPG island, and what you have here are 12 different sites that could be methylated. Of these, some were significantly changed. Site 2 was not, but most of the sites were, and they were changed in the direction of the handled animals having more methylation and probably lower expression of the receptor. This, as I've said, is not exactly what I expected. It's certainly not what the work of Michael Meany and Moshe Ziff would have predicted, but it's very reliable in that it replicates at numerous sites in that system, and always if there's a change, it's always in the same direction of more methylation, more silencing of the gene by a little extra handling. That extra handling gives us something that looks more like a normal prairie vole. So there's lots to do here. The sex differences in this haven't been studied yet, and my last point will be that what I think happens under normal conditions is that when females go through any kind of adversity, they probably have a capacity to put out extra oxytocin. Males may too, but under conditions of adversity, males seem also to be using vasopressin. Vasopressin creates a different kind of strategy, a more vigilant strategy, a more protective strategy, a sort of cliche male-like change. And there's indeed a dynamic interaction between oxytocin and vasopressin under normal conditions. What I think may happen is that females use oxytocin and they have some ability to upregulate that males may not. And this female strategy, which some people have called a kind of passive coping, may be facilitated by oxytocin. Vasopressin more likely plays a role in active coping, being able to energize and mobilize in the face of stress. Now, these kinds of adaptations are very different. Under normal conditions, females have more obligations at times to hold still, to be patient, to deliver babies, to nurse babies. Even the sexual behavior of the female is more of a holding still pattern. Males, a more active pattern. Even with babies, there's some recent work from Ruth Feldman in Israel showing that when babies are manipulated by fathers, it's a much more active interaction. In both cases, actually, she found oxytocin released, but under different conditions in the two sexes. So there's a great deal to understand here. What is oxytocin? Is it kind of a metaphor for safety or social support with healing properties? I think so. Is it a component of the parasympathetic nervous system? Yes, and we have a lot of interesting possibilities to study that. However, it's also a compound that's become part of obstetrics. And it can facilitate, induce labor. It does indeed seem to reduce postpartum bleeding. But what's it doing to the baby? We really don't know much about it at all. It's not just a female sex hormone. Both sexes rely on normal oxytocin. It plays a role probably in both sexes in down-regulating stress, and the healing properties are found in both sexes. It may also make vulnerabilities that allow us to have what people in the department we now work in call mental illness. Processes like schizophrenia and autism, where there are atypical patterns of oxytocin, again, not always in the direction we expect. What's the message? The message is probably that we really need to study this. We really need to understand what we're doing. And the cow's already out of the barn, okay? We're involved in the experiment in humans as we speak because of all the things that I mentioned and all the things that many of you are very concerned about. We are probably working with the safety system, with the social support system, with the love system. We're doing things to it we just don't understand. It can be given and is given in birth as an injection. It can be used as a nasal spray. It turns out, actually, that the stuff that's used for inducing birth is synthetic oxytocin, which is probably the same, but it also contains a preservative to give it shelf life. Little bottles of this are only $2. I started buying them through the pharmacy to use them in animals. Very inexpensive because it has a long shelf life. But we don't even know, we know nothing about what this stuff is doing yet. We're pumping it into moms in the birth process. And if you weren't happy with all that, we can give you some, to end on a little lighter note. You can get liquid trust, and we can spray it around the room, or we would never want to recommend spraying it intranasally, but people obviously are. They're buying it and using it for different kinds of processes. And you probably can't read all the promises here, but it'll reduce stress. If you have any problems relaxing, sleeping, or so forth, everything's going to be wonderful. And it's only 2 for $24, 2 months supply. So what is it? It's an amazing chemical with broad effects on physiology and behavior in both sexes and during development. What it is not, it's not a substitute for love. It's not very well understood. It's not something to be treated casually. And we know very little about effects on other systems, including, for example, the immune system, where oxytocin plays a big role. It may be useful. It may have some uses in medicine. I don't know. I'm very anti-medicine in general, and I would not advise any of you to try this on your kids, but I can tell you thousands of people are doing it already with autistic children. We're also involved, as I've said, in the birth experiment. We're altering the neuroendocrine autonomic system. We have almost no knowledge of it. At the same time, by studying oxytocin, we're beginning to see a kind of what I would call a metaphorical Rosetta Stone for understanding natural healing. And I think we really need to study it. I just am very concerned that things are happening at such a rate that we know very, very little about what we're doing. And I thank you for your attention. I'd love to take questions.
Any comments or questions? Please speak into the microphone so your question is recorded. There's 2 microphones here, and there's one right here. Go ahead. Oh, here's the microphone.
I work with a lot of different families, and I recently had a conversation with a dad who was in a gay married man who already had 1 2-year-old, and he and his partner were about to adopt this sibling of that 2-year-old. And I was just talking to him about oxytocin and how, nursing mothers might have a natural advantage in connecting with their babies. And he said, where can I get that stuff?
Yes, exactly.
Because I don't really always feel like I'm connecting with the baby. Mm-hmm. What would you say to that?
Well, all we have to go with scientifically are the studies that Ruth Feldman's just been doing. And what they found was that fathers playing with their babies were releasing oxytocin. The male and female blood levels, I didn't go through this, are about the same in both sexes. Now, women actually are varying across the cycle, but they're varying around roughly the same mean that the male has. So you can assure them he has oxytocin. The problem is he may also have vasopressin, and the vasopressin, it can actually facilitate social bonds, but it may make those bonds occur in a bit different way, a way that, if you will, is more stressful, more vigilant, more protective. So I can assure you that the father of my children, we have two, was extremely engaged with the children, but at a level of anxiety that I didn't feel. I kind of knew, because I suspect because I was bathed in all this oxytocin, that everything was going to be all right. Steve was not always convinced. I mean, he had to worry. He had things to do. He had to, be sure the territory was okay and the food was there, and he did his part. I mean, he survived. But it's a very good question. I think calm social interactions are helpful, but as I've said, that's not what the natural fathers, at least in the Israeli study, were doing. They were kind of taking the babies up and, laughing and shaking them and throwing them around, holding them over their heads. So that's not bad. That's good. But it's just different. Sorry, I don't have a better answer. Yes?
You mentioned that you thought that the synthetic form of oxytocin was probably okay.
Well,
and I can't cite any
No, I
research,
didn't say it was okay. I just said it was similar to the
Similar.
oxytocin structurally. We have no idea if it's working in the same
My
way.
understanding is that it can block the endogenous
Well,
oxytocin.
any excess amount of oxytocin can block, or any hormone, to my knowledge. I don't know. There may be, I want to be very accurate here. Very small amounts of extra oxytocin may actually feed forward under safe conditions and allow the body to make more oxytocin. But as a general rule, if you give excess of something, the body will cut back its own production. It will also downregulate its receptors. Okay? And this may be some of what we're seeing in the epigenetic studies. It is very hard for me to believe that we are smarter than Mother Nature. Okay? And I don't need clapping for that. It's just obvious. We aren't very bright. We've been at this whole business of manipulating birth for almost exactly 100 years. From the minute they found oxytocin in pituitary extracts, they started giving it to animals and then to humans before it even been synthesized. Then it was one of the first molecules synthesized, so people had access to it in a way that they didn't to most other chemicals, and especially most other brain chemicals. Remember, the main source here is the brain. So it seems to me very unlikely that giving extra oxytocin in the long run will have beneficial effects. That's my bottom line to it. Just because those natural systems are going to adapt. And so at some point there's a price to be paid for the extra chemical, with the body having to now compensate or go back. Now the good news, the optimistic news here, and we really have to hope that I'm right, is that these systems do readapt pretty quickly. And it's not, we don't know that this is lifetime damage. We don't know what we're doing because we've never done a single study, even though it's going on at this amazing level of almost 100% of families. But it's being used in larger and larger amounts. So if you saw my dose-response study, you see that there's good reason to be concerned. More is not better here. Maybe very small amounts, which we hope the body's placenta is keeping it from the baby, but that's another story because the placenta becomes impaired during birth, and we can't be sure if you keep pumping it in that it's not getting there. Anyway, I really want to study this. Anyone who wants to help me, come along. But it's not part of our culture to even worry about it at this point. There was another question, or did you?
I just wanted to comment that in the world of childbirth, when we're seeing increased inductions,
right,
with Pitocin, and then which doubles their risk for cesarean section.
Right.
I'm not sure if it's the oxytocin, the anesthesia, or the combination, but it's inhibiting the production of milk.
Right.
These mothers are having a harder time to breastfeed. They don't stick with it long enough because it's too hard.
Too hard.
Their body's not really working properly. So it's just a cascade of
Right.
effects from this practice.
I agree with you, but we don't have data. There's one study that I know of that came out about 6 months ago from Sweden where they showed that actually Pitocin alone, without an epidural, was associated the next day in the mothers with slightly higher levels of oxytocin. But if there was epidural plus Pitocin, it was quite low, lower than any of their other people. Now, this is a small study. It's not, it's in a journal called Breastfeeding Medicine. It hasn't made the front page anyplace. And maybe it really does need to be repeated, and we need to understand what's going on. And we need to go past one day. What about 6 months later? Is there a change? And so forth. So, again, I think we have to stay optimistic because we're an incredibly resilient weed, as Dr. Sumi calls us. We're a weed species. We can survive and endure and repair and restore. But starting life, in my opinion, with doing things that don't need to be done is never a very good idea. Was there another question there? Yeah.
Can you make it a quick question?
Yeah.
Well, this is a question I've had for a while as I've read on the effects of touch, and what no one seems to mention, when they say the experimenters handled the rats and there were positive effects, and I'm sure there were, but it's one species touching another species.
Yeah.
And to me, it's like that's not, what we need to study. We need to study what one species does within itself,
Right.
Because when we're studying touch on infants, we don't have a dog come in and lick them or a chimpanzee come in and hold them.
No.
So, why isn't that distinction made? And maybe, it's unfair to put that all
I
on
think
you.
your point's really well taken, and we actually did some human-human massage studies. One of the nursing students from UIC used this as her thesis, and we measured it for her, the oxytocin changes. And it went up after massage significantly, but the people sitting in a chair reading got the exact same effect. And this was a crossover. The same people tested twice. And actually, to be honest, the data on touch is not very compelling for oxytocin being selectively increased. Okay? Being safe, feeling good may be good. However, a new study came out of UCLA about 2 months ago, and what they showed, and these, I think they were all men, was that vasopressin actually went down. So they did not see a change in oxytocin at all, but they saw a decline in vasopressin. And vasopressin is, again, a piece of the HPA axis. So what they may have been doing is not necessarily maybe it was upregulating oxytocin and we just don't detect it, but it was possibly helping to regulate and buffer stress. The people, by the way, who got the massage were much happier than the ones reading the National Geographic. Trust me. They felt more relaxed, but oxytocin, and even the Swedes who've really pioneered this, and Uvna Smoberg, hasn't published anything showing a convincing change in oxytocin. So what you read on the internet, let me tell you, almost everything on the internet about oxytocin is either made up or ass backward, okay? And so you kind of have to take it for what it's worth, and if you really want to know something, you better go look for the original data, and it's hard to find. It is, first, most people can't measure oxytocin. Even if they can, taking it out of blood may not tell you what's going on in the brain, and so forth. So there's a tremendous amount that needs to be done. We don't have any developmental parameters. We don't know. In Steve Sumi's data, he and I have a dialogue that starts about 12 years ago. Every time we see each other, I say something like, when am I going to get those bloods? And he says, call me. And then we sort of chase each other around. But one of these days, we're going to look at the monkeys, because my prediction is, based on what we're seeing in animals, it's actually going to be higher in the abused or neglected animals. That would be my prediction. I think the body is coming in and trying to cope with these things, and probably in many ways very successful. But if we block that system, then we're really in trouble. Have we got the question? Okay.
Thank you very much.
Thank you.